Greetings, chem students. This is Mr. Braun, and today we're going to talk about the pre-lab for pH and color indicators. Now, what you need to do is you need to obtain your 96 well plate. That is the one that has the smaller wells in them. It is 12 on the top by 8 running down. So we have the 96 well plate, and you're going to need to obtain 3 pipettes. I'll have the pipettes sitting on the back table. We are going to do a procedure called stretching the pipettes. What stretching a pipette will do is it will give us a smaller size of a drop. If we do not stretch the pipette, we will overflow these small well plates. So we need to stretch the pipette to make smaller drops. You will be stretching 3 pipettes, and you will be labeling them. You'll have 1 pipette for hydrochloric acid, you'll have 1 pipette for sodium hydroxide, and you'll have a 3rd pipette for distilled water. Before the lab occurs, I will show you the procedure on how to stretch a pipette. It's pretty easy to do, okay? Now, it is really important that you keep track which pipette is which, so I'm going to have permanent markers on the back table so that you can label your acid pipette, which is the HCl, your base pipette, which is the NH or NaOH, and your distilled water pipette. Now, this whole procedure of color indicators, we're going to need to make a serial dilution. Okay, you can see right here it says serial dilution procedure. If you follow these directions, it's pretty easy to do. First of all, in A1, C1, and E1, we put 11 drops of the 0.1 molar hydrochloric acid. In A12, C12, and E12, we put 11 drops of 0.01 molar NaOH. And finally, in A2 through A11, C2 through C11, E2 through E11, we put 9 drops of distilled water. You're going to use those to make your serial dilutions. If you follow the directions, it is nice and easy to do because it says, take the entire contents in A1, drop the entire contents in A1, squeeze 1 drop into A2, then put the rest back into A1. So, empty the contents of A1, filling up your pipette, move over to A2, which already has 9 drops of distilled water in it, put 1 drop of what's in your pipette, and then squeeze the rest back into A1. That's the procedure you follow for all of them, except for you just keep moving down the row, okay? So the second one, you'll draw up the entire contents of A2, squeeze 1 drop into A3, and then put the rest back into A2. You'll follow that all the way until you get up to A6. Then if you follow the directions, you use the other pipette, the NaOH, and you start with the contents of A12, 1 drop in A11, and then the rest back in E12. So we are making a serial dilution, and all of those should have different concentrations. All right, so those different concentrations of the acid or the base will allow our different indicators to react. Now, once you get done with your serial dilution, you have to do the same thing for row C and row E. So you're going to do the serial dilution procedure up here for A, C, and E. And when you're done with that, we are going to add the color indicators. So column 1 should have a pH of 1. Column 2 should have a pH of 2, all the way down to column 12, which should have a pH of 12. In each well in row A, so all of these wells right here, you are going to add 1 drop of universal indicator. All of row C, here, you're going to add 1 drop of phenolphthalein indicator. And in row E, you're going to put an indicator of your choice. So it can be one of these indicators right here. They'll be back in the back of the room somewhere, probably on a cart in the back. So you'll use one of these indicators, and you'll have those for row E. Now, when you are finished with all of that, you will have different colors, or they could be the same colors, but there you should have some colors on all of these well plates. So get colored pencils. You should have colored pencils. If not, I have some up in the front. Get colored pencils and color the wells exactly how you see them. All right? Exactly how you see them, and you should be good to go. That's what I will be looking at when I grade this particular lab. I'll be checking how your rows look and what colors they are to see if they match the actual indicator that you have used. Okay? So, this is Mr. Braun. We've been discussing the lab pH and color indicators. Chem 1 students, let's investigate.